Hi, my name is Mary Park. It is week 6, lab 9. And so this is the environmental influence on microbial growth. So the experiment, I would need to incubate the S. cerevisiae and S. epidermidis culture broths containing different salt concentrations to determine an approximate osmolarity that range for each microbe. So with this, like I said before, I have already had completed the actual test already. And so right now I'm going to show you exactly what materials will be needed to do this results. So I'm going to show you right now for the salt tolerance testing. So what is needed is obviously your tubes, and you may want to make sure that you do label them according to the S. epidermidis and S. cerevisiae. You want to make sure you label it, and you want to make sure you label obviously the 3 tubes with the actual S. cerevisiae, and you want to make sure you put 1%, 7%, and 15%. Same thing with the S. epidermidis, the same thing that you would want to do. Then you also want to put 2 drops of S. cerevisiae culture broth in each of the tube, which is the 1%, the 5%, and the 15%. And the same thing you would want to do is 2 drops of S. epidermidis culture broth into the 1%, 7%, and 15%. And then we're going to go ahead and inoculate both of these tubes, all 6 tubes, in my actual designated area, which is the pantry in a box, and I would need to incubate it for 24 hours. So after the observations, after 24 hours for growth, you want to check for turbid. If no growth is observed, then incubate for an additional 24 hours. Observe all the tubes, nothing that the turbidity of the broth, a clear broth, is a negative test. In exercise 2, I will be preparing for the pH broth. With the pH testing, that's something that I would need to incubate the S. cerevisiae and S. epidermidis culture broths with differing pH values to determine on an approximate pH range for each microbe. The pH would range from pH 5.7 or pH 5 to pH 7 to pH 9, and pH 7 being the normal pH. So this is how the tubes would look like when you do label them to pH 5, 7, and 9, and this is for the S. epidermidis and S. cerevisiae for 5, 7, and 9. I would need to exactly add 1 drop of HCl to the pH 5 for both pH 5 of S. epi and S. cere. And then I would also need to add exactly one drop of NaOH, which is right here. These are the actual what I would need to drop into these vials to the pH 9 of both S. epi and S. cere. pH 7 broth is already adjusted to the pH 7 and require no addition of acid or base. The 2 drops of active S. epidermidis to the culture broth into each of the 3 S. epidermidis. Then I would add 2 drops of active S. cerevisiae culture to 3 tubes of S. cerevisiae, which is these tubes right here, into these tubes right here. Then I would need to incubate them for a good 24 hours. My observations will include after 24 hours of growth appear of turbid. If no growth is observed, incubate for additional 24 hours. Then I will need to compare the level of turbid in the incubated tubes. So in my next video, I'm going to show you what the results. In my next video, I'm going to show you the results of my experiment of the environmental influences on microbial growth. Thank you.